Have you ever noticed your gums bleeding for no obvious reason? Or suffered from chronic bad breath that won't go away? Maybe you felt deep, agonizing pain in the gums that you cannot locate? Or notice your teeth shifting, moving, or falling out without any apparent cause? Perhaps you took the next step and went to the dentist, only to be informed that you have bone loss around the teeth. Unfortunately, chances are you have periodontitis. Periodontitis is a multifactorial chronic inflammatory disease that, if not treated, will lead to irreversible destruction to the tissue supporting the tooth, including the bone. Periodontitis is alarmingly prevalent, affecting 90% of the global population, with 20 to 24% suffers from the severe form. Scientific evidence established that the initiator of the disease are the bacteria and the microbe present in the dental plaque that build up over the tooth surface throughout the day. We usually remove it by toothbrushing, flossing, and other oral hygiene measures. But here is what's curious about periodontitis. Some people neglect their oral care and they never get it, while others with the optimum oral hygiene measures, they still get it. So what's the cause of this difference? The answer lies in our genes. Genetic susceptibility to periodontitis is influenced by variation in the DNA sequence called single nucleotide polymorphism, or SNPs. A SNP is a mutation in a single base of the DNA that makes some people prone to the disease more than the others. Interestingly, it varies between population and ethnic groups. So, intrigued by the diverse and complex genetic makeup of the South African population, I decided to conduct a genetic analysis as my PhD study to identify which SNPs, if any, makes the South African population of the Western Cape province more susceptible to the disease. It was a case control study in which I analyzed 24 SNP linked to periodontitis inflammatory biomarkers. The results were fascinating. I identified SNPs in two pro-inflammatory proteins, TNF-alpha and interleukin-1 beta. However, surprisingly, they were preventive against the disease and inflammation. On the other hand, I identified a SNP in OBG, which is a key bone resorption regulator, and this time it increased the susceptibility to the disease 20 times. This genetic profile is unique to the South African population, and it paved the way to the development of novel diagnostic tool that identifies people at risk, and the implementation of precision dentistry, in which the management is tailored according to the individual's needs and genetic variation. Thank you very much.